Well, we're getting thinner and thinner, I think. Okay, so we're starting to lose people, especially the biologists. But actually, I think there's a convention today that's probably the reason. So, have you did some of you look at this
I
article?
looked at it, but I didn't finish.
Okay. Someone else?
I looked at it, I only started.
Okay.
I
So
got to the middle.
yeah, so it's totally okay. I said on purpose. I said this is a recommended article. First of all, because I sent you this article because of the question that arises, and also that you have a possibility to see it like in the future. In general, this is not the greatest review article. In general, as a review, I want to talk to you a little bit about why it's not great. But if you didn't read it, so I will not tell you why it's not great. No, basically, it's not that great because the person that wrote the article, Francisco Bezzanilla, so he did what a lot of times a lot of reviewers are doing, which is focus on specifically what he is doing, and not actually reviewing the whole field. So this was like in this the article started with this figure that shows like 4 possible different mechanisms at which a certain protein can sense membrane voltage, and
And then only one spot.
but eventually it just focuses on one, and specifically also in one type of protein that he is the one that studies. But that doesn't matter. In general, we can get like several concepts from out of this article, and we'll just review them very quickly. Is that you have these 4 different possibilities of how a membrane protein can, or the sensing, voltage sensing mechanism that a membrane protein can have. One of them is very similar to the one that we studied about the sodium voltage-gated channel, that you have these helical formations that have charged amino acids in them, and they can move according to the difference in membrane potential, and thus undergo a conformational change. The second type of mechanism is if you have a specific type of amino acid that has a side group that can change its orientation. Actually, what's changing here are these hedron angles that we talked about in the Ranchana results like a long time ago, about the conformation or the change that certain amino acids can undergo. So this is an example of one of these. Is this conformational change can again change the structure of the protein. Another mechanism that I'm going to go is that if you have this helix that has positively charged residues here and negatively charged residues here, then it will do this paddle motion upon depolarization or changing of the membrane potential. And the last one, which is also kind of interesting, which I think that these two are much less common, but with this one actually it says that the sensing of the voltage is done through an ionic interaction. So if you add, for example, a positive ion, and this example here that fits into this crest or this cavity inside the protein, then it can interact with the residues inside the protein, and once you have the difference in the potential, then this ion gets out or changes the location inside the protein, thus leading to a lack of interaction and maybe a conformational change. So you can have the sensing through the ion and not through the intrinsic properties properties of the.
Like maybe I didn't understand what I read. In some part, he told us about these residues that are either acidic or like basic, and I didn't understand why acidic or basic means that they should react to voltage.
Well, very good question. Acidic and basic are are general. You can use them as general terms to describe the charge. So most of the time, acidic means that it has a negative charge, because also it can absorb a proton, which a proton is a positive charge, it's positively charged. And basic is the other way around, that it readily releases a proton, so it's positively charged. So.
But that, I mean, by the definitions of acidic and basic, that doesn't always imply that acid will be negatively charged and basic will be positively charged.
Yeah, but normally they are. Normally acids, like if you define, you can have an acid that's neutral, but normally when you talk about, and also when you talk about acid and base, you need to say at which range of pH they are also. Some molecules act as acids and bases, like we'll see in a minute, in different pH. So it's just a definition of how we define like the amino acids. You can say acidic or basic, or you can say charged and uncharged, but it really depends on the type of amino acid that we're talking about. Okay, so I will skip this. In general, I would like to talk to you about several technologies that are in this article and several measurements that are made in this article. And the thing is, what you need to understand is that in biology, especially in structural biology, because everything is very, very small, and everything is very, very, you can't do like direct imaging of most components here. You need to do these experiments that you can deduce from their results, the structure or the function of the protein. Okay, so you can do crystallography, but this is really hard. And again, a lot of times crystallography doesn't tell you a lot about the function of the actual protein. So one of the ways to determine, for example, how many charged residues are moved or are translocated from one side to the other side of the protein upon depolarization or change of the protein membrane of the membrane potential, then is like estimation of gating current, and this is the concept of gating current that he talks a lot about the article about this concept. And this is like, let's say you have a protein that, and this is just a very simple illustration. You have a protein that has like positively charged residues that are around here. This is the blue spots. So here the internal solution has to normally supplies two negatively charged ions or charges that cancel out these two residues. And when you have, and let's say we're now switching the voltage and changing the polarity of the voltage, then what you will have is a transient current that results from the movement of the charged residues from this location to this location, thus causing the external solution to contribute to negatively charged particles or charges to cancel out these two over here. And you can measure this small transient current. And measuring this small transient current that occurs very rapidly is called the gating current. So you can some channels have what he gave in an example of the article, gating currents that are 13 electrons. Some have 6, 7 electrons. So it actually implies, and you can also measure the kinetics and the driving force of the potential or voltage sensing element inside the protein with this type of measurement. Another technology that he uses that he describes in this article, this I took not from this article, but an article that this guy published. So it's actually a method that he more or less invented or something like that. Not a lot of people are using it. So it was really like, and he really used this in like a central example in the article. But in general, what's going on here? The research question here is to know which residues of the voltage sensing helix are exposed to the external to external medium from both sides. Which ones are exposed and which are not exposed to the external medium, both intracellular and extracellular. So what the method of histidine scanning relies on the fact that histidine, the histidine residue, can act both. If it has low pH, then it accepts protons, like we said before, and in high pH, then it gives up protons. Okay, it really gives up protons. So here it would be acidic, and here the form it would be basic. So what does it actually contribute to this special quality of this amino acid? Is that you can, let's say you suspect that this residue over here is exposed. You want to see if it's exposed or non-exposed after conformational change of the protein to the outside solution or outside extracellular medium or something like that. So what you do, you mutate the amino acid in this position, and this is the position, and you change it to a histidine. Okay, and now you have four possibilities that can occur. And what you do is that you submerge the cell or the membrane that you want to look at, and you alter the pH, that it will be inside the cell you have a basic pH, and outside of the cell you have a acidic pH. And what will happen when you change the voltage, you will have this conformational change. And if this residue is due to the conformational change is sticking out to the extracellular space, then you will see a current that results from the absorption of protons that are absorbed to this histidine group over here. So you change the voltage and you have absorption. And if the histidine is not exposed, then you don't have any change in the current.
Which one of the two figures on the upper right, which one is histidine? Like, they're both histidine?
Both.
And one is...
One is at pH 4 and one is pH 12. So you see this amine group over here that has a proton in this case, and it doesn't have a proton in this case.
So maybe I don't understand something basic about acidic and basic. What does that mean? Is it acidic or is it basic?
So the pH just means the concentration of protons in general. So if you put the histidine in a solution where there's high concentration of protons, which means it's acidic, which is this pH, we just define 7 or 7.2 as the border of neutrality. Okay, so it's just a definition because that's how our body is in this pH of our body. But but in general, this will be acidic pH, meaning we have high concentration of hydrogen ions, and because this amino acid will absorb protons. So because it absorbs protons, it reduces the amount of protons that you have in the solution, thus making it less acidic. So it's a base. Okay? But really, if you look at just at the molecular, if you look at the atomic level, then it's easier to understand the difference between a base and an acid in that sense. Because here it's sometimes a base, sometimes an acid. But in general, if you have a material that you add into the solution, and it causes the solution to be more basic, then it's a base. Okay? And if you take this, for example, if you take this amino acid and dump it into this pH 4, it will elevate the pH. But if you have a solution that has pH 12, and you dump this material inside the solution, then what will happen is that it will give away. For example, if it has hydrogen, okay, it will give away the hydrogen, thus elevating the concentration of hydrogen in the solution and making it more acidic. Okay, so in this case it will be an acid. But in general, that's also the term that we didn't talk about strong and weak acids. This will be considered a weak acid. Okay, there are acids that are so acidic that it almost doesn't matter if they will always give away a proton. Like just if you take them to pH 1 or pH like extreme pHs, then they will not be considered acidic like HCl, and materials like that. Okay, so I don't want to get too much into it, again, because you didn't read the article, probably it will be hard to understand. But out of these four possibilities, you can either have the residue that's not sticking out at all, it doesn't matter which voltage you're in. You can have a residue that is sticking out just in one voltage, and you can have a residue that in both conformations of a protein is exposed to the extracellular, not to the extracellular, but to outside the membrane, outside the borders of the membrane, thus making it available to, in this case, it will absorb protons, in this case, it will give away protons. And the third method, which is maybe the most important and relevant in biology and also for today, is something that's called FRET. Did you ever hear about it? FRET? Did you hear about fluorescent protein? Okay, so the Forster resonance energy transfer method actually uses the fact that if you have two fluorophores that are very close to one another, if you excite one of the fluorophores, there can be an energy transfer between the two fluorophores, and you actually get an emission in the wavelength spectrum of the second protein, or the second fluorophore over there. So if these two fluorophores of proteins, in general, this is TSP and this is YSP, if these two are very, very close to one another, meaning touching each other, then you will have, if you excite in 414, you will get emission in 525. But if these two are not in close proximity to each other, then you will just get the Kayan emission, the normal Kayan emission, which is 475. Okay. So according to the wavelength that you get when you excite this system, you can know if these two proteins are in close proximity with one another. And here you have an example in this article that you use it to study proximity between different components of the protein or different subunits of the same protein, like we'll see later. Can you remember another method that was used for proximity estimation that we talked about?
Replacing with the cysteine.
Yeah. So the cysteine example that we learned about in the NMDA paper, you remember that they changed and they called this the disulfide bridge, this covalent bond that only occurred if these two elements were very close to one another. Here, it's because these proteins are very large, then it's used only in specific proximity, normally between different subunits and not inside the same
Are
molecule.
they already in the protein, or you have to?
You add them. It's like we'll learn more about genetic techniques in the end of the semester, and then it will be like more clear how exactly you do these experiments. Okay. So last time we almost finished the chapter about transport of ions, and I want to clarify something. Just a minute. I think I told you that what we talked about the K channel or the K passive channel is a type of uniporter. It is actually not the type of the passive ion channel is actually not depicted here anywhere. So just there wasn't an example for that in this illustration in the book. There should be like another cartoon here showing like a normal channel that just doesn't have any conformational change or anything. The difference between that and the transporter and the uniporter is that all these transporters undergo conformational change, and the passive K channel or the passive or any passive ion channel doesn't undergo conformational change upon binding. However, these do. In the symporters, antiporters, and even the uniporters, the energy from the moving down the gradient of this molecule induces the conformational change also of a unipolar. Okay, so basically the only channel that we talked about that doesn't undergo conformational change are these passive ion channels that we showed the example of the potassium passive ion channel, which we call the resting K, and it's not even depicted here, but all of these channels are actually undergoing conformational change. It still doesn't say that all of them are active. Still, these two are passive because they don't use the energy of one molecule to transport the other one, and so they're still considered like these two passive, but they're considered active.
Did we give an example of an actual unipolar?
No. A lot of time unipointers are used for transport of glucose and materials like that. And normally we don't talk, in this course we don't talk so much about metabolism, so I don't think that we're going to see, maybe no, I don't think we're going to see an example of a unipointer. Okay. So, the last segment in this chapter regards neurotransmitters and the receptors, and also transport proteins and the role they play in signal transmission in synapses. So you learned about the neurotransmitters probably, right? So you know that like which neurotransmitter here is unique? Like in a sense, it has like almost all. Okay, so maybe it's like not a good question that I'm asking, but molecularly or atomically, all neurotransmitter except acetylcholine are derivatives of amino acids. Okay, so glycine is just glycine. Glutamate is glutamate. Which what's special about glutamate? You know?
It's almost an amino acid. It just has a little change from glutamic acid.
Oh, but that's the function of glutamate. What is it?
Metabolic.
Right. This is the major excitatory neurotransmitter of the body, in the brain. Dopamine, norepinephrine, and epinephrine. Epinephrine is actually adrenaline, so just the scientific name of adrenaline. So they're all derivatives of tyrosine. Serotonin is a derivative of tryptophan. Histamine from histidine, and aminobutyric acid or GABA is another derivative of glutamate. And what's special about this? Right. So this is a major inhibitory neurotransmitter, and all the other ones, sometimes they're termed neuromodulators, sometimes they're termed neurotransmitters. So in the field in general, there's a lot of debate about what is regarded as a neuromodulator and a neurotransmitter. Like if we're talking about serotonin, for example, or acetylcholine or dopamine, a lot of times they can function locally as a neurotransmitter, but also they can function as what you would term a neuromodulator. That normally it says that the function is more spread out and more like the neuromodulator is just there floating around the cells and modifying and modulating their activity, and not like present locally at the synapse or released locally and fast in the synapse. But so it's like a not well defined term, the term neuromodulation, and you'll see that in a bit. So how did a neurotransmitter actually get packed, and how does this mechanism occur? So in general, you need to understand that each neuron synthesizes or produces just one type of neurotransmitter. This is like biologically, generally, okay, obviously there are exceptions, but in most cases, what defines a neuron as cholinergic or GABAergic or glutamatergic is the neurotransmitter that it produces. But, and it's a very important point to understand that a cholinergic neuron can have GABAergic receptors, it can have dopaminergic receptors, it can have glutamatergic receptors, and all of them together. So a a neuron that senses, for example, acetylcholine is not cholinergic neuron. Cholinergic neuron is the neuron that synthesizes acetylcholine. Okay?
I have a question.
Yeah.
So, for example, one neuron can get input from many neurons, right? So it can have like many different receptors.
Yeah.
Ah,
It expresses a large variety of receptors. Okay, and you can get from one inhibitory neuron. For example, the simplest example is that some synapses are inhibitory, some are excitatory. So the type of receptors that you have in each one of the synapses is different. Okay. So these vesicles are the mechanism of action of how these vesicles are filled with neurotransmitters is by the function of a V-class proton pump. Okay, and these are also ATPases. Again, when we say pump, we usually mean a protein that uses ATP and the hydrolysis of ATP in order to move a substance against the concentration gradient. So again, you can see here again the example of the pH. So if you have high concentration, you have lower pH, and this gradient of hydrogen is then used with an antiporter, with a specific antiporter that fills the synaptic vesicle with the neurotransmitter, with a specific neurotransmitter.
But
So
how are the vesicles with a high concentration of ions created in the first place?
so they're created with you mean how the vesicle itself, or how they're filled with hydrogen?
How they're filled with hydrogen?
With exactly with the V ATPase pump. Okay, like the example here. This is hydrogen. Okay, these molecules are hydrogen, and they're pumped outside from inside the cell to the lumen of the vesicle. Okay. So in general, what do you think about this process? How does it look to you?
Too complex.
Why not? Yeah. Why does the cell
Why
just
two phases?
doesn't invent this type of channel that just passes that takes ATP and moves in this example like acetylcholine or the neurotransmitter against its concentration gradient and fills the lumen? Why not? Can you think about maybe one type of explanation? If you're thinking like biologist and you're thinking about, let's say you are so in general thinking about logic in biology is not a very good idea, but you can try to get in the logic. Some would say the logic of the designer, and some would say the logic of the system. But in general, it's like I like to think a lot of the time of the cell as a mathematician. Okay, if you have a way to get to a solution, then why invent the wheel over and over again? Okay, so you have these proton pumps, and you can use the same mechanism. You just have to change this antiporter, which normally is a very small and non-complex protein, and you just have to change this molecule between all the different types of cells. And this mechanism can be conserved like in all the types of the cells, and just has to be specialized in moving hydrogen. Why do you need to invent if you already have a mechanism of pumping in hydrogen? Then you can just use this mechanism in order to in a variety of cells, and you just have to change this antiporter here. Okay, to use the potential energy that is stored over here in order to pump the neurotransmitter inside the vesicle. Okay? It's just one hypothesis. It doesn't mean that this is the reason. Okay? Another reason, maybe, can be, if you remember, the fact that not all processors are acting at the same speed. Okay? So the ATP pumps are the slowest pumps that we have, or the slowest way to transport ions. And the transporters can be 2 to 3 orders of magnitude more or faster than this ATP. So, and also a lot of times in the cell, we see a phenomenon that you have that you have different populations. Why is it moving in the opposite direction? Okay. A lot of times you have this phenomenon where you see that the vesicles are not are kept in different conditions. So, for example, you can have many vesicles that are just pumped with hydrogen, waiting to be pumped a neurotransmitter inside, and you can also have vesicles that are neurotransmitter, and then you can have, but we'll talk about this, maybe we'll talk about this in a second.
So
Okay.
what, I mean, why do you do that to the hydrogen? I think I didn't understand how can you do it.
Why am I showing here the hydrogen part?
No, just why do you put hydrogen inside?
Because the hydrogen is acting as a concentration gradient or energy source, because the hydrogen concentration here is much higher than outside or in the cell. Okay? So the cell can harness this energy from this concentration gradient, from the difference in concentration, and it harnesses it by taking energy from protons that it will now move with the concentration gradient of the proton, and use this energy in order to pump, or to not pump, but to move neurotransmitters against their concentration gradient. Okay, so this is a classic antiporter that uses that one molecule goes with concentration. One molecule goes with the concentration gradient. Okay, this is the hydrogen in our example, and this is the neurotransmitter that is being transported against the concentration gradient.
Okay, you could just transport the neurotransmitter.
Yeah, so that's what why I propose that some kind of mislogic
Okay,
of
but
the
the
cell.
pump was not. Okay, so I thought you meant like to the synapse.
No. This
Straight to the synapse. Okay.
this is
So
not
I
in the synapse. This is just packing. This is just packing the vesicles filled with the neurotransmitter. So these vesicles, actually, this whole process is happening in close proximity to the synapse, and this leads me to maybe I talked before about the.
It doesn't happen in the soma. We learned that it happens in the soma and travels to the synapse.
Well, yes and no. Okay. So in general, that's part of the concepts in biology that people didn't know, so they assumed certain stuff. Okay, but in general, a lot of the packing of neurotransmitter occurs in the synapse. Okay. Obviously, also many of this packing is done like in the cytosol or close to the soma, but you'll see in a minute that this process happens a lot in the. When we talk about what happens in the soma, normally people mean that the neurotransmitter is produced in the soma and then gets shipped, but the packing into synaptic vesicles a lot of time happens locally. Okay, but what you're talking about is kind of a transition in thought that neuroscience is going through in the past years about local procedures that were thought to happen in the soma, but now we understand that they happen closer to the synapse.
So the neurotransmitter production does happen in the soma, or is it
I don't
also?
I don't want to say. Okay, so it's not. I want to teach you only the basic concept. So in general, I will not say if it's produced in the soma or produced somewhere else, but it's produced in the cell. Okay. So
I have another question.
yeah.
You said that the pump is slower than the antiporter,
Yeah.
but it doesn't really matter. I mean, in order for a neurotransmitter to be packed in a vesicle, first the pump has to work, and then the antiporter. So you're still like your slowest player is the one that causes the speed.
Okay, we'll answer that in a second. Okay, remember that question. So a little bit about metabolism of neurotransmitters, and like we discussed before. In the example of acetylcholine, you have the enzyme that manufactures acetylcholine from acetyl-CoA and choline. It's called choline acetyltransferase, and this is the enzyme that produces the mature neurotransmitter. And interestingly, in the case of the cholinergic system, the antiporter that actually pumps the acetylcholine inside the vesicle, genomically, and now I return to a little bit what we talked about genes. It's contained. The gene for the antiporter is contained inside the gene of the enzyme of the choline acetyltransferase. Okay, and what do you mean that contained inside? showed you this image before about the about the processing of an mRNA. So in general, I would like to just emphasize that you have, if this is the DNA and this is the genomic sequence of an mRNA, then initially this whole molecule is being transcribed, okay, to what we call pre-mRNA, and in this state it has exons, which are segments that will eventually be translated, or will be apparent, or will appear in the mature mRNA, and you have introns that already inside the nucleus, when the mRNA is inside the nucleus, will get spliced out. So we call this process splicing. And these introns, and here it's very misleading, because normally introns are much larger than exons. Okay, so here they look like small segments, but they're actually, most of the time they're very large relative to the actual exons. So what happens in the example that I showed you here is that this is a plot from a like online very used tool that you can visualize genes with. So this is just the coordinates of the genome, and in chromosome 10, and here you see the CHAP, which is the choline gene, and each one of these thick lines, the small thick lines here, are exons, and the thin lines over here are introns. Okay, so this gene actually has 16 exons and 15 introns, and as you can see, the introns are much longer than the exons. And what you can see is that inside the first intron, contained inside the first intron, you have this mini gene, which is the SLC18A3, but its friends call it VACT. So it's a vesicular acetylcholine transporter, or vesicular acetylcholine antiporter. So it's actually, the information for this gene is contained inside the intron, inside a segment that is not used by the host gene, but in this gene it actually produces, it has the information for the gene itself, for the mini gene itself. And this gene specifically doesn't have any introns, okay? Just like one consecutive axon.
How does it, like, how does the cell know to translate it afterwards?
Okay, so what happens? It can happen through many pathways, many different options. One option is that initially you just have the full mRNA. This large piece is being transcribed to RNA, and then after the splicing, after this segment is being cut out, it's being once again edited and converted to be a mature mRNA of its own. Okay, so you can have like one gene is garbage, and the other gene is the actual material for the other gene. So if we're talking about a mechanism like that, then you have a very close relationship and a very close regulation between these two genes that are very much linked in their biochemical pathways. Okay, but you can also have independent promoter regions that you will get specifically just this gene without this gene, and also you can get this gene and nobody tells us if they're maybe like after the splicing it just gets thrown away. Okay, so they can also be just independently regulated. Okay, but the answer is probably both. Okay, so we know that there is independent promoter regions for this gene also, but also that many of the copies of the gene arise from the mature molecule, the large molecule that is like its host gene. Like a lot of times you say the term is host gene. So the CHAP gene is a host gene for the SLC18A3 gene. Okay? And this mechanism is highly conserved. Like you see this in almost all organisms that have a cholinergic system.
So you're saying there's another place where this gene is also in the genome, it just doesn't always get transcribed, or this is
No,
the only
this is the only
Okay.
location in the genome that you have this gene. Okay? But I'm saying that it's conserved among different species. Okay? In evolution. So after the vesicle gets pumped with neurotransmitters, with neurotransmitters, then this vesicle has causes the fusion of this membrane? You know?
CA2 influx.
Right. Okay? And not depolarization. Okay? The depolarization itself is not what causes the synaptic, the vesicle release over here. What senses the protein that senses that depolarization is a calcium-dependent voltage gated channel.
But is
Not
the
calcium-dependent, sorry.
depolarization just non-directly?
Yeah, not directly, but it's important. Okay? Because you can have like the specific processes and blockers that can block like one type of process, not the other. For example, you can if for some reason you have like permeability to calcium, or you have this leak of calcium, then you can also have synaptic release without neurotransmitter, and and also different processes. So I didn't mean to say calcium dependent. I meant to say voltage dependent calcium channel. Okay, that senses the change in the voltage, introduces calcium inside the synaptic region, and through the aid of these very specialized snare proteins, which are here, causes the fusion, and this fusion we term as exocytosis. Okay, so there are two major ways that we talk about fusion and inclusion of membranes to other membranes. The process where a membrane fuses with another membrane and releases a substance to the extracellular space is called exocytosis, and the reverse process in which the vesicle is being pinched in, like we say, it's called endocytosis. Okay? So we'll just go over a little in a more orderly way about on this process. So first you have import of neurotransmitter, again through the use of hydrogen gradient. You have the vesicles that are pumped with neurotransmitter. Normally they're in the cell we divide it into two subpopulations. We normally say that there's a vesicle store that is filled with neurotransmitter, but it's not docked to the synaptic cleft or to the synaptic region, and it's like waiting in line for them to dock. And then you have the second population of synaptic vesicles that are fused to the synaptic cleft or to the synaptic membrane, and are waiting for an activation signal to cause this cascade and this fusion. Okay, so once you have depolarization that arrives, and the calcium channel detects this depolarization, introduces a wave of calcium inside the synapse, and this calcium is what causes the fusion of the membrane with the.
And how the calcium does that?
We're not getting into that, but basically you can imagine. Do you remember that we saw these proteins that can sense calcium or can bind calcium? So a lot of times the binding of the calcium undergoes introduces a conformational change. Like
So there are like calcium receptors in the vesicle?
they're not receptors. They're not considered like receptors in general, but they're calcium-sensitive proteins. They're called snare proteins. If you want, you can read more about it, but we're not getting in so much into the detail of the actual mechanism. It's actually pretty complex. Like you have like many proteins that are involved in this cascade, and upon the binding of calcium, it induces the effect of the fusion of the two membranes. Okay, of the membrane of the vesicle and the membrane of the membrane of the cell. Then you have the spitting out of the neurotransmitter. But because also many of you have come, I think, from psychology and psychological background, you know so after the synaptic release, you have two mechanisms that are related to the recycling of this whole process. One of them is reuptake. Okay, so the neurotransmitter is getting. Have someone haven't heard about the process of reuptake?
I learned about it within lab.
Okay, so actually, almost all neurotransmitter except, and we'll talk about it, except acetylcholine, again, especially in this sense, the termination of the signal, the synaptic transmission, is through large neurotransmitter back into the synaptic cleft, where again it could be repacked close to the synapse. So from here, the neurotransmitter is not like it's traveling all the way back to the soma and then getting packed and then moving back. Okay, so a lot of the packing is done, is being recycled in the close to the synaptic cleft. And the process in which this the vesicle is being recycled, because you can't have so much membrane fusing all the time, and you have to it will cause like the membrane to expand, and normally the size of the membrane doesn't expand because you have to release. So you always have these two processes that are happening simultaneously. Some of the membranes are getting are fusing, and some of the in other locations or in proximal locations, membrane are getting included or endocytosed. And this is done through the help of a very, very special huge protein, and actually a protein complex that is composed of many of these proteins. This is actually a very close to life model of this clathrin, and this clathrin cage is what actually facilitates the inclusion of the vesicle. So the vesicle is being formed or endocytosed into this structure, and later the structure is just disassembled, and you have a vesicle that is ready to be repacked with hydrogen and then with neurotransmitter. Question? Okay, so just to show that it's not that simple, last time I want to show you like that, like how real biology looks like, not these cartoons. So I found a movie showing a model of actually how the synaptic vesicle.
This model represents a cutaway view of a synaptic vesicle, with the membrane lipids and proteins drawn to scale. Each synaptic vesicle membrane contains approximately 7,000 phospholipid molecules and 5,700 cholesterol molecules. Each vesicle also contains close to 50 different integral membrane protein molecules, which vary widely in their relative abundance. The most abundant protein is a snare protein, called V-snare. This molecule participates in membrane fusion at the synaptic terminal, and there are about 70 copies of this protein per vesicle. By contrast, the vesicle contains only 1 to 2 copies of V-ATPase. The V-ATPase uses energy from ATP hydrolysis to pump protons into the vesicle lumen. The resulting electrochemical gradient provides the energy to import neurotransmitter molecules, such as glutamate, into the vesicle. Through a transporter in the membrane, protons flow down their concentration gradient to the outside of the vesicle, as glutamate enters by an antiport mechanism. In this way, a vesicle is loaded with thousands of glutamate molecules. This model was created in part from an electron tomogram of a real synaptic vesicle. The tomogram combines visual slices of the vesicle at various angles to create a unified image. Other data, including structural data of the synaptic vesicle's proteins, have been combined with the data from the tomogram to create the three-dimensional model on the left. Note, only 70% of the membrane proteins estimated to be present in the membrane are depicted in the model. A real vesicle would be covered by an even more dense forest of proteins.
So you can understand that a vesicle doesn't look like this smooth, nice membrane. Okay, everything is filled with many, many proteins and undergo enormous amount of regulation in general. And just to conclude, talking about the question that you asked before about what does it matter if you have like more ATP pumps or more transporters. So you need to think about it like that not. And really, we're talking about the central nervous system and the cortex specifically. Many, many cells are not firing all the time. Okay, so a lot of time the firing patterns between different cells can be very different. So you can have very little amount of ATP but the ATP is active all the time. Okay, so the ATP is like a packing facility that just packs vesicles all the time. And these vesicles are stored aside and they're waiting for do not have to be linearly linked with one another.
Why can't we store vesicles with neurotransmitters? I mean, I understand why there's a different mechanism for creating the vesicles and for fusing them with the membrane, but we could just have one pump or one molecule that fills them with neurotransmitters, and then when they're needed outside, there's a different mechanism that fuses them with the membrane.
Well, again, first of all, you're right. We don't know the question. Like in any sense about the biological reasoning, a lot of time what we think is not logical. A lot of time we found out that it is logical, and we just didn't understand many of the processes. But a lot of times also there is no logic. And it's just like I told you before. It's just because certain mechanisms were involved in evolution, and certain solutions were found to problems in evolution, and thus the cell is like easier and more in the sense of parsimony or simplicity. It's easier to group together a group of solutions and not make a specialized solution for a specific process. Okay, so we'll take a break and we'll continue after this.
כי בעצם בתוך הווסיקולה אין נוירוטרנסמיטר, אז מבחינת הגרדיאנט הנוירוטרנסמיטר כן ירצה להיכנס, אז למה צריך את ה-A? כאילו לא הבנתי את הכיוונים.
אז העניין הזה, מה שאת שואלת זו שאלה שהיא מאוד טובה וקשורה קצת יותר מודינמית. זאת אומרת, אם אנחנו עכשיו רוצים הריכוז, העניין שהריכוז היחסי של הנוירוטרנסמיטר מחוץ לתא הוא יחסית לא מאוד גבוה. זאת אומרת, זה לא שהפנים שהתא מפוצץ בנוירוטרנסמיטר, ויש לנו מלא נוירוטרנסמיטר, ועכשיו אנחנו רק צריכים לפתוח תעלה והנוירוטרנסמיטר עף פנימה. זאת אומרת, הריכוז של נוירוטרנסמיטר גם מחוץ לתעלה הוא די נמוך. אז אנחנו חייבים איזשהו מנגנון אקטיבי שיודע סלקטיבית לקחת את הנוירוטרנסמיטר ופשוט לדחוף אותו. זה נכון שזה כאילו נגד הריכוז, אבל הוא בעיקר חשוב מתי שהריכוז של הנוירוטרנסמיטר באזור של הווסיקולה מתחיל לעלות. מתי שהוא כבר מתחיל לעלות, אז כבר מתפתח גרדיאנט. לפני זה, את יכולה לחשוב על זה, זאת אומרת, יש שם עניין של גרדיאנט יחסית. זאת אומרת, אם אנחנו מסתכלים, אם זה התא, ויש פה איזה וסיקולה, אז באופן כללי יש לנו מעט נוירוטרנסמיטר, והריכוז היחסי, אז גם יכול להיות שבתהליך של איזשהו, שבהתחלה אתה לא צריך כל כך הרבה אנרגיה, זאת אומרת, האנטיפלוטר לא ממש כל כך אנרגיה, אבל הוא לא הגיע למצב שיש פה ריכוז גבוה של נוירוטרנסמיטר, שזה מה שאנחנו רוצים. ומה שיקרה, זה שרק מתי שנוירוטרנסמיטר יתחיל להיות מוזרם באופן אקטיבי, אז הריכוז פה יתחיל לעלות, ואז אתה באמת צריך לדחוף כנגד הגרדיאנט. פשוט הנפח היחסי פה הוא מאוד מאוד קטן, אז מבחינת בשביל להשוות ריכוזים לחוץ, מספיק לך מעט מאוד נוירוטרנסמיטר בפנים, אבל אתה רוצה בעצם להגיע לרמה הרבה יותר גבוהה של נוירוטרנסמיטר.
כאילו בהתחלה זה בעצם כביכול עם נוכל ריכוזים, אבל באיזשהו שלב זה יהיה להפך.
כן, מאוד מהר זה יהיה. זאת אומרת, כמה מולקולות...
בגלל שהכמות בחוץ מאוד קטנה, והיחס של הווסיקולה לגודל של התא מאוד קטן.
כן, כי אם הריכוז היה פה באמת מאוד מאוד גבוה, נגיד כמו שאנחנו מדברים על איונים, על פרוטונים של מימן, אז שם באמת יש הרבה יותר, ובגלל זה הרבה פעמים גם הריכוז זה משמש יותר תפקיד. נגיד, אם אנחנו מדברים, אולי הדוגמה יותר טובה בדיונים, נגיד שנמצאים מחוץ לתא, כמו פוטסיום שנמצא כאילו פה, או אשלגן שנמצא פה, או נתרן שנמצא בחוץ, אז שם באמת הריכוז, בגלל שגם הנפח, גם של הפנים של התא וגם של החוץ של התא הם יחסית משמעותיים, אז בהגדיר ריכוזים כל כך משמעותי, זאת אומרת זה יותר מ-0 גודל, אז באמת יש עניין של דחיפה לאחד הכיוונים. זאת אומרת, ברגע שאתה פותח תעלה שהיא לא ספציפית, שהיא פשוט מאפשרת זרימה חופשית, אז יש לך זרם של איונים שיזרום לאחד הכיוונים. אבל פה זה גם מתווך על ידי הפוטנציאל החשמלי.
תודה.
צריך עוד זה? זה יש עוד משהו?
לא יודעת. יש לי איזו תהייה, זה לא כאילו סתם שתמיד אותי.
כן.
היאפטק זה על ידי
זה על ידי סימפול, אבל אנחנו נדבר על זה עוד שנייה.
כאילו כל הכוח הזה שמה יותר יעיל, נראה לי משהו מוזר, כי הייתי צריכה לא להשתמש בו בפסיק פשוט להעיף את זה החוצה, לא?
לא ממש חושבת על זה שזה צריך להיות דיגיטלי. זאת אומרת, הסימולציה צריך להיות כמה שיותר דיגיטלי. אם את רוצה להעביר, חושב שאת רוצה להעביר, נגיד סיגנל שהוא פשוט on and off. אם את רוצה להעביר סיגנל שהוא פתאום קורה ופתאום מפסיק, אז את צריכה מנגנון שישחרר בבת אחת כמות עצומה של נוירוטרנסמיטר ועד יפסיק. כי אם את מדברת על שחרור, אז יהיה פשוט מין שחרור כזה פסיבי וכל המנגנון הזה יהיה הרבה יותר איטי. כי גם אנחנו מדברים על שחרור של 5,000 מולקולות של נוירוטרנסמיטר בבת אחת. לעומת מצב שאם את צריכה להעביר אותו בתוך תעלה למשל או משהו כזה, אז זה יהיה מצב הרבה יותר איטי. זאת אומרת, הריכוז היחסי יהיה הרבה יותר איטי.
אוקיי, תודה.
אבל שוב, זה גם, מגרן זה לא תראה לי מגרן. So just
Okay. Yeah. Okay.
just to show you again a more real life example of how synaptic. Sorry. Alright. Okay, just to show an example image of how a synaptic cleft looks like. So you can see here that again you see the formation of many vesicles that are not fused to the actual synaptic cleft, and you also see a vesicle here that is we can't know if it's being extracytosed or endocytosed, but in general you can get the general the feeling of the distances also that we're talking about in the synaptic cleft. One of the questions that I would ask is why again I'm going to the questions of why, which is a mistake, but I would just say that what I think is the advantage in this form of synaptic release is that you get, and again you need to understand the distances here. The distances here are really small molecularly. So the distance between the synaptic cleft is about 200. It ranges from 200 nanometers to 200 angstrom, which is 20 nanometer. And this distance is actually facilitates very fast diffusion rates. What we normally think about as like a material that diffuses, it's like a slow process, but actually when you talk about very short distances, it's a process that happens really fast. Okay? So, and if you have this massive synaptic release, what it gives the neuron is two things. One, it allows it to elevate the concentration of neurotransmitter here very fast. And the second thing is to do so, and this is a lot of time not regarded a lot, but it also allows to control the size of the response. Okay, so you can have all the synaptic vesicles that are linked here to be released, or you can have some of them get released. And a lot of time you have specific types of modulations, like the size of the action potential normally doesn't change. Okay, but a lot of times if you have some kind of modulation that can alter the concentration or the permeability for calcium, then it can trigger like a more massive synaptic release. Okay, in response to stimulus. So this mechanism is allow a fast response and also the ability to modulate. Okay, the ability to control. And so the way that and I'm not getting into that because I'm assuming that we're going to talk about it in more different courses. The way that this signal is getting terminated, the synaptic signal is terminated, is by two main ways or two main mechanisms. The most common way is through reuptake, in which normally a sodium powered SIM porter that introduces sodium together with the recycled neurotransmitter inside the synaptic cleft. Okay, so this is a sodium SIM porter. And normally, together with the sodium, there's also entrance of calcium in order to balance the electrical potential of the sodium. The sodium is a positively charged ion, and the calcium is negatively charged ion.
You mean chloride? Calcium is...
No, sorry, chloride. Chloride, not calcium. It's written Cl. So. Chloride, yeah. So almost all neurotransmitters except acetylcholine are being reuptake, and acetylcholine you also have reuptake, but only one component of the molecule, and actually the termination of the signal is done through a special enzyme. It's called acetylcholinesterase. Okay, that is a very fast enzyme, one of the fastest enzymes in nature. And it hydrolyzes the acetylcholine to acetate and choline, and later the choline is being transported back to the presynaptic site. So you have reuptake, but not of the whole neurotransmitter. Okay, and in general, these reuptake mechanisms are a lot of times targets of many therapeutic drugs, or non-therapeutic drugs, or general drugs. So cocaine is a very potent dopamine reuptake inhibitor. fluoxetine or Prozac is a serotonin reuptake inhibitor, and Ritalin is both a norepinephrine reuptake inhibitor and a dopamine reuptake inhibitor. Okay, so what these drugs cause is actually they elongate the time, or they extend the time that the neurotransmitter is in the synaptic cell. Okay, and we're not going to talk about the effects of that. So in this, we'll start talking now about a segment about channels in general, or ligand-gated channels. And when we talk about neurotransmitter receptors, they fall into two basic categories or classes. The first one is a ligand-gated ion channel, meaning that the receptor for the neurotransmitter is a channel by itself. Okay? And this is normally called ionotropic, and also the mechanism of action is much faster. And once you have a ligand that binds, then it immediately opens the channel. And the second type, which we'll start talking about today, is G protein-coupled receptors. That the binding of the neurotransmitter or a single molecule induces the opening and closing of a separate ion channel that's not in the same protein. But this is actually a very simplistic way to show it, because G protein-coupled receptors do a lot more stuff than just opening channels, like through a second messenger or something like that. And we learned about it that they're in charge of multiple processes that happen inside the cell. And these are the G protein are a subclass of metabotropic receptors. Okay. So we'll give a very simple example of a ionotropic channel and a receptor, which functions at a very specialized synapse called the neuromuscular junction. Okay? And this synapse, and here you can see on the electron microscope scan or image, that you see the axon terminal of the presynaptic cell, the presynaptic neuron, and this is the membrane of the muscle cell that's being activated. That will get activated, and you'll learn a little bit about this in physiology, I think, with Mickey, about and this is the sarcomere, or the unit that contracts in the muscle cell. So, how does the synapse actually activate or act? Well, the neurotransmitter in this synapse is acetylcholine, and here it's strictly a neurotransmitter and not a neuromodulator in any way. And the mechanism of action is through a channel or a receptor, and it's called nicotinic acetylcholine receptor, which is shown here. And once you have an external signal or action potential that reaches the synaptic terminal, again you get an influx of calcium due to opening of voltage-gated calcium channels, and a synaptic release of acetylcholine to the synaptic clefts. So, about each vesicle that carries acetylcholine in the synapse contains about 3,000 or 1,500, depends how you look at it, can result in opening of about 3,000 ion channels over here. And the main permeability of this channel is to sodium. So after the sodium, after two acetylcholine molecules bind to this receptor, you have a large influx of sodium, but still this influx of sodium is normally not enough to cause the muscle to contract and to so what you need is an amplification signal that is normally mediated by voltage-gated sodium channels, similar to the ones we studied about in action potentials, and they cause an even more dramatic influx of sodium inside the cell, which depolarizes the cell and causes the release, you don't see it here, but causes the release of intracellular calcium, and this calcium is actually what causes the contraction. Okay, the contraction of the muscle. So, looking in more detail to this receptor, so it's a pentameric protein. It's assembled out of 5 subunits. 2 of them are similar, the alpha 1 subunits, the other one are different. This is an semi-simulation, semi-data from specialized imaging about how the spore actually looks like if we're looking at it from the top. And actually, this channel is permeable both to sodium and both to potassium. Okay, but because the cell or the muscle cell is normally very close to the reversal potential of the potassium, then there's only minor permeability or minor amount of potassium ions that actually pass through this channel. And contrast to what we learned about the resting K channel, in this channel, both ions are pass through the pore with their water cage, and they're not hydrated, as we saw in the example of the selectivity filter of the potassium channel. So this channel also has a very specialized structure that are, you can say, built in order for them to attract positive ions, which is this high concentration of negatively charged amino acids that are located both, you can say, the entrance and the exit, but it doesn't really matter because the motion is bidirectional. And these negative pockets are actually help the channel to attract positive ions. And as I said before, you need to have two acetylcholine molecules bound to this channel in order for it to open. Okay, and they do this. Again, we're returning to concepts that we talked a little bit about at the beginning of the year, and they bind in a cooperatively. They bind positively cooperatively, which means that I gave the example of the hemoglobin protein. That once you have a binding of one molecule, then the possibility of binding the probability of binding a second molecule is increased. Okay, so it's positively cooperative. One interesting fact is that if you get bit by a cobra, the actual mechanism that will make you die is through the binding, very, very tight binding, much tighter than the actual neurotransmitter to the site where the neurotransmitter normally bound, and here it bounds to all 5 subunits. And this binding is actually very, very strong, and which eventually leads to paralysis, and when it gets to the heart, you die. Okay, so don't get bit by a cobra. It was a joke, not you're too serious. Okay. So for that, we conclude the part about transport of ions and molecules across the cell. And we'll start the very last chapter, which I think I want to shrink a little bit, but we'll see how it goes, about G-protein-coupled receptors. And we're going to start with a general review or general overview on signaling molecules and cell surface receptors. So I hope you all know that we are a multicellular organism. Okay, we're not an amoeba, and we're not like a sponge or something like that in the sea. And our cells need to communicate with one another. Okay, so opposed to what normally most neuroscientists think, not the only source of way of communication between two cells is through a synapse. Okay, we can have many ways of communication, especially when we're talking about development, it's especially important. And this communication is done through a group that is called signaling molecules or cell surface, and is mediated by cell surface receptors, meaning just receptors that are obviously on the surface of the cell. So here we have an example of the 7 major cellular pathways that are controlled by external signal, and G-protein-coupled receptor is just one of them, and we're going to focus on that. But before we focus on G-protein-coupled receptors, we're going to, like I told you, we're going to we're going to get acquainted with several terms about signal transduction and signal molecules. So, in general, there isn't one category of molecule that is a signal molecule. Almost all types of the biological molecules that we talked about can act as a signal. Can act as a signal molecule. It involves like amino acids, lipids, neurotransmitters, neuromodulators, peptides, and proteins. All of them can function as signal molecules, and these molecules bind when we refer to them as ligands because they bind to the receptors, and they do so with, again, a wide range of ionic interactions, including ionic, van der Waals, and hydrophobic interactions. And in general, the term that is this, like these cascades over here, are normally termed signal transduction, meaning the propagation of the signal, what the external signal causes, and what exactly is the biological process that is affected from the signal. So, if we're talking about we'll first talk about the first part of the signal transduction, which is the signal molecules binding themselves. And we can divide these molecules into three categories. You have, according to the distance in which they act, meaning that endocrine molecules that are normally, we term them as hormones, act on distal regions, meaning that they're normally carried in the blood or carried in the CSF or any type of biological fluid, and they affect, most of the time they affect cells and they affect processes that are further from where they were produced actually. Okay? CSF, cerebrospinal fluid. You didn't learn about the stuff the brain is in, floating? Okay, so maybe we'll talk about it a little bit, but I think we want to talk about it in physiology, so I'll just say a few words, because it's important. The cell is not like the medium in which the brain is submerged in or surrounded in. It's called the CSF, the cerebrospinal fluid. It's not like blood or something like that. It's transparent. And it's very well isolated from the blood. Okay? It's not that the brain doesn't get a lot of blood cells that are going inside the brain, but the barrier between the medium that the brain is in and the blood cells is a lot of times called the blood-brain barrier, that you a lot of times you've probably heard about. And this is what differentiates. So the CSF is all over like inside the skull and along the spinal cord. It's actually what defines the central nervous system. Is submerged in CSF. Okay. For example, like what we said before, like this medium over here, if we're talking about a cell, like a neuron, this is CSF. Okay, the extracellular medium is CSF. So, again, we talked about endocrine. Paracrine signaling is what you would, inside this category of paracrine signaling, meaning that molecules that affect target cells that are in close proximity with. So, synaptic transmission is a subtype of paracrine signaling. And you also have autocrine signaling, which in cell response to substances that they release. Okay? So, again, I'm not going to get into that about the logic of this, but in general, it can have many purposes in the cell to sense its own molecules that it itself is releasing. Okay? And also, you can have some signaling molecules that can act both in the short range and the large range. And the long range. For example, epinephrine or adrenaline can act as a neurotransmitter and a hormone. So, and especially during development, these signaling molecules, or the differences in concentration of these signaling molecules, are actually the key factors that determines to which organ or to which cellular subtypes the cell will eventually differentiate into. Okay? And the most classic example of this concentration is taken from Drosophila, in which this is the neural tube. You will learn a little bit in development, I think, also in physiology, about this structure. But this is what will eventually be the nervous system of the fly. And according to the different concentration of a special signal molecule, in this case, a protein, which is called Sonic the Hedgehog, and it's really the name of it. I don't know if you know Sonic the Hedgehog. Maybe you're too young, but a lot of times when you hear like funny names, we also learn about the tabby gene and or sort of the derivatives of that. And when you hear these funny names, it's normally because it was discovered by in Drosophila. Because the Drosophila people, contrast to other scientists, have a sense of humor, and they normally call like we talked about the if you remember, we talked about the potassium channel, the shaker K channel. So it's called shaker because the mutation in this channel causes the fly to do this shaking motion. Okay, so this is one of the reasons. And in a minute I'll show you what a mutation the Sonic the Hedgehog does and why it's called Sonic the Hedgehog. But in general, what's interesting about it is that you can get the feeling about that a simple concentration gradient, meaning that if the cells over here feel this concentration, the cells here feel this concentration, it causes the cell to differentiate into an entirely different type of cell. Okay? So it's not only the signaling molecule, but the concentration of the molecule, which is very important. And obviously you can imagine that there are many, many more concentration gradients that are existing in parallel to this concentration gradient. So you can have many molecules that are in this axis, and also in the z-axis. And the combination of these signaling molecules a lot of times determines the set of genes, for example, that this cell will express, and it will determine the fate of this cell and the differentiation of this cell. So this is just from a nice review that I took from Nature that's showing how with simple gradients you can have boundaries. As you know, like in the brain, we have many structures that are very strictly bound. Okay, so you have sometimes structures are more vague, but sometimes the brain structures like the amygdala or structures similar to that are very well defined. Okay, and how this definition and how this region gets like to have these barriers is in this article they show a very simple example of how this process can occur. So it starts with a simple gradient, and then there is like a threshold here, like a threshold that you can see here, that if you have a high concentration, so cells that saw a higher concentration turned into these cells, and cells that saw a low concentration turned into these cells. And then there is like a process, and after the what we call an endocrine signal, what we said like normally this is like a molecule that is secreted by some kind of external organ and is in charge of this gradient. Then you start to have local signals, or what we call paracrine signals, that the cells themselves are transmitting to one another because they sense certain cells around them. So there starts to be like this chain reaction, combinatorial chain reaction, that after these cells initially differentiate into this specific type of cells, they start releasing their own set of molecules. And for example, a cell that is sitting here will not get the same combination of signaling molecules like a cell that's sitting here, because here a cell will get molecules from this cell and from this cell. Okay, so the combination of these two factors a lot of times causes the cells in the gap between the two cells to differentiate into a third type of cell, and this third type of cell can also affect the cells around it, and vice versa, and it's a very complex reaction. This is very simplified opposed to what really happens. So you're asking why. I know that you're dying to know how a fly that has a mutation in Sonic the Hedgehog looks like. So it looks like something like this. I don't know if you've ever seen an actual fly, but it doesn't look like this. For example, it doesn't have wings. Okay, it has like this small stabilizing wings, but it has this porcupine structure with its hair and all sorts of really, really bad developmental issues. Okay, and normally these flies don't live so long at all. moving on from the molecules themselves, the actual reception, or the way that the receptors act, normally is that you have that a certain receptor or a certain type of a receptor is very specific, meaning that it binds to only a specific type of molecule. But you can have but a specific type of molecule, a signaling molecule, can bind to multiple receptors. Okay, so many receptors can are activated by acetylcholine. For example, muscle contraction via ligand-gated ion channel, like we saw before, or G protein-coupled receptors that act in slow heart rate, and also in the digestive system, it's through a third pathway, and everything is the same, regulated by the same molecule. But in specificity terms, and here we see the example of the growth hormone and its receptor. So first of all, you can see that a lot of time, that's also one of the things that I'm putting emphasis in this course, is to understand like proportion of things. Okay, like normally you would say a receptor is obviously much larger than its molecule of its ligand, right? Because you learned the example from an enzyme and stuff like that. You see these, right? You see these caricatures of a channel, and you have this small ligand that's binding to it, and it's called conformational change. Well, it doesn't have to be like that at all. And in this example, the actual the protein is a lot of times larger than the actual receptor, or the ligand in this case, which is the growth hormone, is much larger than its receptor. So this is just to show like this tight binding site, or this very specific binding site between the growth hormone and the growth hormone receptor. Okay, so moving on to the actual effect of the signal transduction, or the next effect of the signal transduction. After the molecule is being recepted by the specific type of receptor, it can have, in general, two effects. Okay, it can either change the activity of existing proteins, or alter the activity of existing proteins, or it can change the amount of a protein being produced. This is, again, like I told you, is especially common in development, which a lot of times this cascade ends in a specific production of a set of genes. Okay, so this cascade can get to the nucleus and cause transcription of specific genes. And this is like the second that change the amount of protein that is produced by a cell. Specifically, when we're talking about G protein-coupled receptors, they can do both. Okay, so they can change the activity of a protein, or cause a downstream reaction that will introduce more proteins. So another concept that is important about these signal transduction is that some classes of receptors can initiate multiple pathways. Okay, so we can have, let's say, acetylcholine bind to a certain receptor, so it doesn't necessarily have to induce a certain pathway. It can activate multiple pathways. But a lot of time, what we see in the cells is that that different pathways a lot of times act through in what we call this second messengers, and I will elaborate about this in a second. So these pathways act through very specific subtypes of molecules, meaning that you can have like a lot of different types of receptors and a lot of different types of the signaling molecules, but in the end, what it comes down to is that cellularly, like in the cell, the molecules that mediate the response are normally the same. Okay? So there are generally four common molecules which we will term as second messengers. And why are they second messengers? Because the ligand molecule that activates the pathway is the first messenger. Okay? So the second molecule in line after the activation of the receptor, or what we learn about the G protein-coupled receptor, is normally a small molecule, which is termed the second messenger. And the most common second messengers by far are derivatives of ATP, you can say, which is cyclic AMP. And the reason that it's cyclic, and this is adenosine monophosphate, okay, in contrast to adenosine to the sugar through two connections, which form this cycle. Okay, so the phosphate group is linked both to the 3-carbon and to the 5-carbon of the sugar, forming the cyclic shape. And another type of this cyclic molecule is a cyclic guanosine monophosphate, which which again, these all are derivatives of nucleotides. Okay, this is the A nucleotide and this is the G nucleotide. These are the types I don't want to elaborate so much about, but they're also existent. And one that is very important, and another secondary messenger that is very important for neuronal functioning is calcium. Okay, so for example, if we're looking at the NMDA channel, I'm going to show you in a minute, an NMDA channel is permeable to calcium, and we can actually look at the NMDA channel as a signaling mediator receptor. Okay, that means that if you activate this receptor, you have influx of calcium, and calcium can cause all sorts of things. Okay, and this is like a very small list of actually what calcium mediates. It mediates a lot of cellular responses. But for example, in the muscle, it causes contraction, like we done before. In endocrine cells, it causes exocytosis of secretory vesicles, and in nerve cells, it causes exocytosis of neurotransmitters. Okay? Questions? So, moving on the pathway, what does the secondary messenger actually do? So normally, what the second messenger does is it activates through specialized enzymes that either add phosphate group or remove phosphate group from target proteins. And these are called kinases. Kinases are proteins that add phosphate, and phosphatases are proteins that remove phosphate. And you have two types of the protein kinases. It's just two types of the families of proteins. One that adds phosphate to tyrosine, and one that adds phosphate to serine or threonine residues. Okay, so in general, you need to think about there's a huge family of these kinases, and I don't know if you remember, but when we talked about phosphorylation or modification of proteins, phosphorylation is actually like a switch. So if a protein is phosphorylated, it can either be activated or it can be silenced. So what this cascade does after the signaling molecule is recepted here, then the second messenger is being activated or put into play, and this second messenger normally what it does is activates this group of proteins, and this group of proteins can activate many types of protein or deactivate many types of protein. So you can see that this is like a flow of cascade of a reaction that can be very much that gets amplified during many of the processes, during many of the steps of this cascade. Okay, so just one last point about intracellular signal transduction in general is that many times the different components that are related to a certain pathway are clustered together. For example, in the synapse, we have a very important scaffold protein. We call it like a scaffold protein because it keeps structures, which is called PSD95, or in its full name, postsynaptic density 95, that is highly concentrated in postsynaptic boutons or synapses or around the synaptic spines. And this protein actually interacts with many membrane components. For example, neuroligin. How does it sound to you? What is ligin? Almost, but not exactly. So ligand. Why is it called ligand? A ligand? Because it binds, right?
Okay.
So it's like ligated. When something is ligated, it's bound. Okay. So neuroligin is actually neuroligin is the postsynaptic membrane, and neuroexin, which is its partner protein in the presynaptic membrane, are actually what stabilizes the synapse together. So they're like the two major components, two major And the NMDA receptor and glutamate receptor, you already.
How is PDZ related to what we just spoke about?
The PSD95 or the PDZ? We didn't talk about the PDZ.
Okay,
So the PDZ domain of the PSD95 protein is a specialized domain. It's actually a very not a very how do you say? It can bind to to many proteins because the target sequence is very permissive. Okay, so this PDZ domain is actually the binding site of many membrane proteins, and this is actually this protein is actually, you can think about it as it's keeping all the proteins together and preventing them from diffusion, right? Because we learned about the membrane in general in this fluid mosaic model that you have lateral diffusion of them. So in order to prevent this diffusion, you have these PDZ domains, and when I say that they're not, they're very permissive, they have very permissive binding sequence, meaning that you just need to have a serine or a threonine, any type of amino acid or a hydrophobic amino acid, and you can get binding to this.
But how is this related to signaling?
So this is here in order to show you that different components of signaling. For example, the NMDA receptor that opens that is permeable to calcium is in close proximity, for example, for phosphorylation agents or downstream molecules that are affected by the calcium efflux. Okay, so a lot of times these structures are being held together, and the signaling, the molecules that are related for the signaling pathway are a lot of times held in close proximity to one another. So this whole structure is being anchored to the cytoskeleton, to the actin filaments, through another type of protein that doesn't really matter. Okay, so I think we will stop here, and next time, next class we'll go through all these chapters. First I will explain specifically about G protein-coupled receptors and G proteins in general, and then we'll see examples of G protein-coupled receptors that affect cyclic AMP, or through the enzyme that produces cyclic AMP, which is the cyclase, and G protein-coupled receptors that regulate ion channels, which are very important, obviously. This I don't know what we'll talk about, and this is another example of G protein-coupled receptors that activate gene transcription, which is also a very important mechanism. I think I will give you an article for next time. This time the article is more than recommended. Okay, it's a very basic article by researchers called Kandel, one of the founding fathers of neuroscience, and it relates to topics that we talked about and topics that we're going to talk about in the next class. If you have questions, again, feel free.
אתה מעלה פה מצגות מעודכנות? שיש
אני יכול לעשות. פשוט אני בדרך כלל עובד עליהם, כאילו עושה את השינויים האחרונים, עושה סרטונים, עושים כל מיני דברים כאלה ממש כמה ימים לפני השיעור.
אחרי כאילו השיעור זה גם בסדר, פשוט כי הרבה פעמים אני רושמת לי כזה, להסתכל במצגת וזה, ואז אם המצגות לא תאמות, אז קשה לי כאילו להבין.
אני בכל מקרה, בסוף השנה, בסוף הסמסטר אני אדאג לזה שכל המצגות המוצאות יהיו שמוכנים. אבל אני יכול לעשות.
תודה.
Yeah, that's the problem. That's why I don't want to read it very much.
No, but it's still for me. That's the same thing.
Yeah.
אוי, לא שמתי את זה.
Yay!
fastest way. We're the leading in the social interaction between people in LSEC. I think before we came to LSEC, there was no like all the people were like everybody went into their thing, but the place like you will need each other, not necessarily like now in the courses obviously you need each other, but later you will need each other even more. Like in the cooperation that you will form between the research of each one of you is very, very important. So I would very much recommend like to start to having this meeting. Yeah.
Like what gets
So what are you having, a party?
But this is like a big topic.
No, but this is for reading.
Yeah, this is already in common.
So we're reading like Wednesday, like
Yeah,
yeah.
this week on Wednesday and I thought it on 100 April.
Yeah.
we had the
What this is Thursday?
Yeah. You're probably busy. But I have time to if I slow down.
Only under zero.
I'm just saying you have people to do it.